This program is brought to you by Stanford University. Please visit us at stanford.edu.
Good evening, everyone, and welcome to our session focused on Darwin's legacy in medicine. As usual, there's a great opportunity to talk about Darwin and medicine tomorrow at noon in Building 50, Room 51A. Lunch is really informal and fun, and I hope you'll give it a try. A participant last week came to the Tuesday lunch and sent me an email saying, quote, "I really enjoyed Tuesday's post-lecture discussion, some very engaging stuff, and certainly helps to make the class content resonate." The writer suggested that I should say more about, quote, "the added value of the more informal, dynamic dialogue on Tuesdays," so you have been so reminded. Our speaker next week is Professor Russell Fernald of Stanford's Biology Department, a prominent researcher and dynamic lecturer in the area of brain and behavior. Please remember to sign up for lunch on Tuesday, 11/18 with Russ. Please also remember that important date I told you about last time, January the 5th in the new year at 5 p.m., right here in Cubberley, free, the world premiere reading by professional actors from the Stanford Drama Department of a terrific new screenplay about Darwin's life and all its trials. The reading is offered as a special post-course treat for you with music, special effects, and other things you won't want to miss. Following, there'll be a chance to meet with the playwrights and discuss the screenplay writers and discuss the script with them. Do make note of this special opportunity, January the 5th at 5 o'clock, right here for you. Finally, undergraduates, you'll have to forgive me. I got caught on a deadline of my own. I know you understand deadlines, so the midterms will be back in section on Wednesday rather than tonight. Now, last week, we heard from Stanford professor Melissa Brown, who spoke about Darwin's legacy in the social sciences, as you'll recall. Melissa outlined many good reasons why Darwin has gotten a bum rap in the social sciences, right on up through today. First, she said, Darwin's other big book, The Descent of Man, was not as thoughtfully written as Origin had been, and contained a number of troubling arguments about sex roles, for example, or about indigenous non-Western peoples, and she gave us some examples. Noted Brown, today's social scientists found some of this work appalling. Secondly, she said, Darwinism has become confused with progressivist theories about stages of development in human society, even though Darwin really said nothing of that kind, from the likes of Herbert Spencer, Lewis Henry Morgan, etc. In fact, today when you say evolution in anthropology, more than half of the time, this is what people will assume you're talking about. Stages like from savagery to barbarism to civilization, unilineal scales of progression. A third argument she gave was that Darwinism was also associated via a cousin, Francis Galton, and Herbert Spencer's expression, survival of the fittest, with such offensive rights violations as eugenics and compulsory sterilization laws. With all of these negative associations, it's little wonder that social scientists are skeptical about Darwin. But Brown continued, even some recent attempts to reconcile Darwin and the social sciences have backfired. Professor Brown mentioned sociobiology with its self-proclaimed goal of, quote, biologicizing the social sciences. We hadn't heard that word until sociobiology came along. And the campaign to find a natural selection explanation for most of what humans do. She also mentioned evolutionary psychology, which looks for Darwinian algorithms in the human mind, but winds up with what Professor Brown described as untestable just-so stories about past environments where the algorithms might have been adaptive. In both of these cases, the social sciences simply ask, where's culture in all of this? In the epistemology of sociobiology and evolutionary psychology, there is precious little culture there. The rest of Brown's talk, Monday and at lunch on Tuesday, was about a whole different tack. What about focusing on Darwinism, she said, as a process, as a model of a particular kind of change, and asking whether there are analogous Darwinian processes of change in culture and social systems. In other words, treat them as existing, acknowledge their existence and importance, and ask, is there an analogous Darwinian process of change astir? At lunch, she did a nice job exploring this idea further. She said, quote, let's take culture to be the system of shared meaningful ideas in a society. And let's take social organization to be the hierarchical structure of roles in the society. She proposed that these are both inheritance systems akin to the familiar genetic inheritance, but separate and distinct, existing apart. Instead of genes, the cultural track of inheritance has units that Brown called ideas. That's what Dennett had called memes before her. And the social track of inheritance, she says, has roles, where a role, a social role, is defined by rights, duties, and position within the structure or network. Brown claimed that both culture and social structure may very well show their own version of Darwinian evolution, of variational evolution, that is to say, a culling of variation based on some winnowing process. More ideas are introduced than persist at high frequency in a given cultural system, so our job in the social sciences, she said, should be to figure out which ones persist and why. The persistent ideas are ideas of high cultural staying power or cultural fitness. And roles are often created and elevated in a given social system, so there too social scientists should ask which roles persist and multiply and why. The persistent roles have high staying power, which implies a high fitness in the system of social inheritance. Professor Brown concluded by saying that the strivings of individuals, she feels, heavily influence the staying power of ideas and of roles in their separate realms. Right or wrong, Brown's view of social science shows the full impact of Darwin's legacy via analogous processes, while also avoiding the mistakes of attempts to biologicize the social sciences. Tonight it is a pleasure to have with us a scholar whose ideas about Darwinian medicine and infectious disease have been well received and thus enjoy high cultural fitness, to use Brown's term, in the fields of medicine, epidemiology, and beyond. Dr. Bob Siegel will introduce our speaker tonight. Bob?
Thank you, Bill, and thank you all for persisting with us throughout the quarter. It's a tremendous honor and pleasure to present tonight's speakers and discussants. Not only will you be hearing from three preeminent researchers, but they will be discussing a topic that combines two of my very favorite subjects, Darwin and infectious disease. What could be better than that? I have brought along some of my friends to share in this. We have Ebola, and we have sleeping sickness here. You'll probably hear about Helicobacter pylori, HIV. Oh, of course, we always have this friend, too. Here we go. I got that. And let's see, we also have, rabies. Okay. Anyway. So we should have fun tonight. I even wore my hepatitis B bow tie. Though we are most aware of microorganisms when they invade our body and cause disease, humans have an extremely intimate relationship and interface with the microbial world. As Dr. Rothschild noted, the number of bacterial cells in the human body outnumber the human cells by a factor of 10 to 1. These bacteria provide a host, pun intended, of functions including production of essential vitamins, as well as even protection against more dangerous pathogens. Beyond these vast numbers of colonizers, you also heard that long ago the types of cells that comprise our body, as well as those of plants and animals and protozoa and fungi, so-called eukaryotic cells, entered into a series of symbiotic relationships with bacteria. Such endosymbiotic bacteria now exist in degenerate form inside every cell of our bodies. These chloroplasts and mitochondria now exist as organelles that live with provide the cell with energy machinery in exchange for a comfy environment and everything else they might need. A third example of our microbial symbiosis is the fact that there are retroviruses, distant cousins of HIV, that have integrated into our very DNA instructions. In fact, more of the letters in our 3 billion letter sequence of instructions that make up the human genome code for retroviral sequences than code for human genes. Who knows what additional relationships may remain with microbes to be discovered? Microbes, by virtue of their rapid replication, error-prone reproduction, and large numbers of offspring, can evolve at dazzling rates, even compared to a finch's beak. Rates that conveniently allow us to watch evolution in the wild or even in the test tube. We have heard how genes and culture may be independent replicators affecting human biology and behavior. Tonight we're going to introduce into the mix another group of replicators which may also have a profound impact on humans, namely the numerous and diverse inhabitants of the microbosphere. We will hear about how evolutionary principles can give us deeper insight into infectious disease and even disease with no obvious infectious behavior or character. Our speaker tonight is Dr. Paul W. Ewald, professor of biology and director of the program on disease evolution at the University of Louisville. I learned how to say that today. He also holds a joint appointment in the Department of Microbiology and Immunology at the School of Medicine. Professor Ewald was a founder of the discipline of evolutionary medicine. Dating back to 1980, he's published numerous papers and book chapters on the subject. And he is the sole author of 2 important books. Let's see if we can do this. That work? There we go. Of 2 important books on the subject: The Evolution of Infectious Disease and Plague Time, the New Germ Theory of Disease. Dr. Ewald was the first recipient of the Smithsonian Institution's George E. Birch Fellowship in Theoretical Medicine and Affiliated Sciences. He has also been an important popularizer of the concept of evolutionary medicine, including articles in Natural History magazine and the New York Times. Dr. Ewald's work has been covered extensively in the scientific and popular media, including Newsweek, Fortune, Discover, Scientific American, and many, many others. An article by Judith Hooper states, quote, "As far as Ewald is concerned, Darwin's legacy is the most interesting thing on the planet." He was referring, of course, to this class. What prescience. Dr. Ewald's work is a call to arms to embrace evolutionary theory and change our concepts of infectious disease. This work has important implications not only in predicting and preventing epidemics of acute infections, but also in alerting us to the profound impact of chronic infections, which may progress in an undetected and insidious fashion, ultimately emerging as cancer, heart disease, and other important medical conditions. Conditions that continue to plague us even with the application of a, quote, most advanced medical techniques. This work has considerable implications for the practice of medicine and for economic and public policy. Brandishing evolutionary principles and recalling Pasteur's pithy insight that fortune favors the prepared mind, Dr. Ewald admonishes sciences to approach human diseases open to the critical role of infectious agents. Let's give a very warm welcome to Dr. Ewald.
Okay, well, thank you very much for that very generous introduction, Bob. Today I really want to focus on one particular question. I've got the sort of formal title here that you can read, but really I want to focus on what causes cancer. Now, one would think with all of the research that's been done on cancer that we'd have a pretty good answer to this question, but I'm going to argue that basically what we've had is little parts of the puzzle, and we still don't have a great answer that provides a kind of cohesive conceptual understanding of this question. What I want to emphasize today is that we're really in the middle of a rather quiet revolution in understanding cancer. And that understanding of cancer that quiet revolution has been going on for at least 30 years, and we could trace it much further back than that. And this revolution involves a steadily increasing recognition of the role of infectious causation of cancer. And I would argue that we're right in the middle of this. And sort of the subtext of what I'm going to be talking about today is giving you a sense of my view, at least, of what that revolution is looking like and where it may lead in the next couple of decades. So to get a sense of what I mean by this quiet revolution, I want to take you back to the 1970s, to a time in which I was very malleable based on the kinds of things that were taught to me. I was an undergraduate and then a graduate student in the 1970s. And any of you that are old enough and can think back at that time would hopefully remember that the idea that infections could cause cancer was really kind of a wild idea. It was raised, it's actually been raised throughout the 20th century, but if you look at medical texts, they would typically say something like what was stated in the Cecil textbook of medicine that was published in 1979. To date, there is no unambiguous evidence that any class of human cancers is regularly caused by a virus, and here he really means a pathogen. It could be bacteria, virus, or protozoan. Some cancers have been shown to be associated with elevated levels of antibodies to certain viruses or viral components. However, no direct evidence exists that the viruses and the cancers are causally linked. So this is pretty much the state of conventional thinking back in the 70s. So what I want to do in this sort of quick sweep through the last 30 years is talk about what happened in the literature and in medical thinking during the 3 decades since this statement was published. What I've done here is listed in the left-hand column cancers that now have been accepted as caused by infection. The second column, I've got the pathogens that are accepted as the causes of those cancers. The third column, I have the approximate year accepted. Now, I say approximate here because this acceptance is really quite gradual. It tends to take place over 5 to 10 years. Once the first critical paper is published, there'll be some people that might accept it, but as a whole, medicine hasn't accepted infectious causation of cancer until it's had time to digest evidence, until subsequent studies have been published. So what you can see in that third column is that these cancers that have been accepted over the last 30 years have been accepted fairly steadily. That's why I say it's a sort of slow, quiet revolution that's been going on. 1980, the year after the publication of that last quote, we had Burkitt's lymphoma being accepted. But that was kind of a weird infectious causation because that involved a virus, Epstein-Barr virus, the cause of infectious mononucleosis, together with a protozoan. And when that was accepted, people said, oh, yeah, but that could just be some quirky thing that happens in the tropical areas where you've got this disruptor, Plasmodium falciparum, messing up people's physiology. And the second one that was accepted around 1980, adult T-cell leukemia being caused by human T lymphotropic virus type 1, was something that was sort of distant to most people's experiences, at least in Europe and in the United States. It's not very common in Europe and the United States. It's much more common in sub-Saharan Africa and in some parts of Asia and in the Caribbean. Things really started to change in the mid-'80s when cervical cancer was accepted as caused by certain serotypes of human papillomaviruses. But then since then, we've gotten other cancers being caused by the viruses listed there in the second column. In the last column, I've just given you a brief sense of where these cancers are the most important, but these cancers are not necessarily restricted to those areas. It's just that's where they've been most studied and seem to be most prevalent. Okay, so what you can see is there's been this steady appreciation of infectious causation of cancer by the medical community. What's interesting, at any given point, if you asked medical experts, let's say in 1985, well, how many human cancers are caused by infection? They'd say, oh, 5%. Maybe a little less than 5%, maybe 3%, and all the rest are caused by, mutations, gradually dysregulating cell proliferation. In 1995, you ask the same question, they'd say, oh, maybe 10 to 15%, and all the others are caused by, the standard arguments. So what's been happening is during this time, people who've been arguing against infectious causation of cancers gradually sort of moved away from that position, but almost grudgingly. And the real question is, do we really know what causes the rest of cancer? And I would argue that basically we don't know for most of these cancers, the full story, because the full set of hypotheses haven't been investigated yet. And so that's the other theme that I'm going to be trying to portray to you as I go through the next 45 minutes or so of this lecture. But as of last few years, reliable sources have now come to the conclusion that about 20% of all human cancer is caused by infectious causes. And I'm defining infection very broadly to include parasitic causes as well, things like wormy parasites as well as protozoans. Okay, so this is quite a change, going from essentially 0% accepted as caused by infection to about 20%. And the real critical question is, what about the other 80%? How many of these are actually going to turn out to be caused by infection? It's a very important question, because once we know that a disease is caused by infection, there's almost always something we can do to make major inroads at controlling that disease, because medicine as a whole has proven to be very good at blocking infection by one or another means, maybe by improving hygienic standards like screening blood supply, or by making vaccines against pathogens that cause disease, or sometimes by using anti-infectives to cure infections. So I would argue that's an incredibly important question, and we should try to avoid having an approach that might blind us to the correct answers. So to avoid being blinded, I always like to come back to the basics. I would argue that we can think of causation of disease in the context of a triangle, and each apex of the triangle corresponds to a different category of causation. And I would argue that these are the only three categories of causation of disease. And I can do that by a little bit of hand waving, but I think it's actually a very defendable position. So we have inherited alleles that can either cause a disease or predispose us to a disease. We've got parasitic organisms, again broadly defined to include multicellular parasites, eukaryotes, and subcellular parasites. So I'm including viruses and bacteria and viroids as parasites. And then in the lower left-hand corner, we've got sort of everything else. That was sort of the hand waving. I'm putting everything else in that category that I refer to as non-parasitic or non-infectious environmental causation. This is just too much or too little of something that is not living. Okay, too much of some dietary component could poison us. Too little of it could lead to a vitamin deficiency. Too much radiation could lead to cancer, skin cancer, for example. Too little could lead to insufficient vitamin D synthesis, if we're talking about UV radiation, and so on. Now, I like to present this as a triangle for a kind of heuristic reason, and that is that I think we should envision putting disease entities somewhere in the body of this triangle. And that steers us away from thinking that there's just one cause to a given disease. So if we find that there's one primary cause, a cause that if you eliminate it, you won't get the disease, then we would tend to put the disease close to that category of causation in this triangle. If the other categories of causation are just exacerbating factors, sort of secondary causes, then by putting it not right at the tip of that vertex, but moving it towards the other vertexes to some extent, we're reflecting or we're acknowledging the exacerbating roles of these other causes. So what does this look like in terms of real diseases? So lung cancer, the general dogma is that lung cancer is caused maybe by genetic predispositions together with environmental mutagens. In particular, tobacco smoke is considered the leading cause of lung cancer. And so we would put lung cancer close to the environmental vertex, but we'd have it on that axis there connecting genetic and environmental to emphasize that there can still be genetic predispositions. However, I would argue that just because we found that these are contributing causes, we should be very careful about concluding that parasitic factors are playing no role if they haven't been investigated. This is an error that's commonly made in health sciences and sciences in general. That is, when we find out part of the answer, we sort of jump to the conclusion that that's all there is to it, and then we scoff at hypotheses that might run contrary to the thing that we know is playing a role. We know that smoking is playing an important role in lung cancer. Another cancer is retinoblastoma, and this one is one that's brought forth as an example of how mutations are known to contribute very directly to cancers. And we know that it's about half, a little more than half of the retinoblastomas, these cancers of the retina, are caused by mutations in the retinoblastoma gene when an individual's already inherited one of the mutated copies, and then they just need the other copy to mutate in order to have both copies mutate, because you have to have both copies of the genes altered in order to have retinoblastoma. And a much, much smaller proportion of people are acquiring the mutations within a lifetime. Usually it's inheriting one copy of the mutation. However, because even if you inherit one copy of the mutation, you still have to have mutation caused by some environmental factor. We may not know what it is. We may just say it's sort of random. That doesn't really explain what's causing the mutation. We know that that we should put that cancer somewhere between genetic causation, the inherited causes, and the environmentally induced causes. Okay, and there again, I think almost everybody would say retinoblastoma should be right up against that line, because we've got no evidence that infectious processes are involved. But I would also add that nobody's looked at it. So it could possibly be, although I wouldn't bet any money on it, it could possibly be that infections are playing a role, but it'd have to be some very common infection that almost everybody has in order to explain the association between the mutations and the very strong association between the mutations and the occurrence of the disease. Okay, cervical cancer we think of as cancer caused by infection, but we wouldn't put it right at the apex on the lower right, because infection by itself, in this case infection by the oncogenic serotypes of human papillomaviruses, infection by itself isn't sufficient to cause cervical cancer. You also have to have mutations. And so we would say that viruses are a primary cause of cervical cancer, but so are the mutations. You just have to have both in order to get cervical cancer. So both would be co-primary causes. If you eliminate either one, you probably wouldn't get cervical cancer. So at least tentatively, we can sort of put it smack in between. But by going through this process, you begin to get a sense as to how you're going to avoid mistakes that often have been made, in which somebody finds part of the answer and then they just scoff at another part of the answer, even though that other part of the answer may be true. Okay, so let's take a look at where we've come in terms of understanding the process of oncogenesis. You know, for most of the last 50 years, most experts have really focused on genetic predispositions and then subsequent mutations that eventually lead to metastatic cancer. However, now that we know that infectious agents are playing a role, we have to incorporate them somehow. And the way that this was generally incorporated was by sort of changing the dogma as little as possible. So the dogma was related to mutations. So the way in which people suggested that infections or other or parasitic causes in general were related to cancer was by stimulating mutations. Infections trigger inflammation often, and inflammatory compounds often are reactive, and they're known to be able to cause mutations. So this seemed very reasonable. Another alternative hypothesis is that the infections are a primary cause. Again, we can think of infections possibly as playing such an important role that we would have cancers moved close to the infectious apex, or at least halfway between mutation and parasitism, as I showed for cervical cancer. And that's quite a different idea, the idea that maybe infections get the ball rolling. They're not actually contributing to cancer by causing mutations, but they're doing something fundamentally different. And we can assess this idea, and we can distinguish between those two hypotheses by learning at the molecular level what pathogens are actually doing to the cells when they're playing a role in causing cancer. So a lot of information has been generated, and I think rightly so. A lot of the studies have been focusing on certain barriers to cancer. So pathogens could be breaking down those barriers by causing mutations in the gene that code for the proteins that somehow provide a barrier to cancer, or the pathogens might have actual proteins that they make themselves that interfere with those barriers. Those are fundamentally different kinds of processes, and they bear very strongly on those two alternative hypotheses. Because if the pathogen is producing a protein that interferes with a cancer barrier, then that supports the idea that the pathogens could be getting the ball rolling. And if that's a little confusing, wait till we talk about this table, and I think it'll be very clear. So there are 4 critical barriers to cancer. Cell cycle arrest, which means the cell is kept from dividing. Immortalization of the cell, which means that the cell, instead of having a very fixed number of divisions that it can go through, it could go through an essentially unlimited number of divisions. Apoptosis, which is cell suicide. So when cells are, something's going wrong with the cell, if it's going down the path to cancer or if it's infected, the cells can trigger their own destruction by triggering apoptosis. And then the last barrier is cell adhesion. If cell adhesion is altered, then cells that could be growing uncontrollably could be moving from one spot to another, and that's a key aspect of metastasis. Okay, so we want to understand full-blown metastatic cancer. These are 4 critical barriers. And I put these 4 critical barriers up here because each of these barriers to cancer could be a barrier to a pathogen like a virus that is being selected to live inside of a host. So what do I mean by that? Well, if viruses, from a virus's point of view, the longer it can live inside the host, the better, all else being equal. So if it's living inside of a cell and it's replicating and generating virus particles that go out into the bloodstream, those virus particles are being exposed to the immune system. They may be destroyed fairly effectively by the immune system. But if instead they can live inside of a cell and cause the cell to divide, and they can divide right along with it, they can restrict the exposure of their proteins to the immune system. So they may be able to hide out in cells. So how do they do that? Well, one way in which they could do that would be to stimulate the cells to divide, to break through cell cycle arrest. And on the left-hand column here, I've got several different viruses. These are the viruses that we know the most about in terms of their biology. Among those viruses that are associated with cancer, human cancer. Okay, so I've got sort of the 5 viruses that have probably been that are associated with cancer, believed to be causally associated, accepted as causally associated with cancer, that have been very well studied in terms of their molecular mechanisms. Okay, so let's just take a look at Epstein-Barr virus. This is this virus I mentioned before that causes mononucleosis. And that virus produces proteins that cause the cell to break cell cycle arrest. So I put in this column under cell cycle arrest, yes. Well, what about immortalization? If the virus doesn't interfere with that cap on reproduction, causing the cell to divide is only going to be of limited value because it'll divide and then suddenly it'll get to that cap and it won't divide any longer. But if it could somehow dysregulate the protein that's responsible for breaking through that cap. This is telomerase, for those of you that know some of the biochemistry inside of the cell. If the telomerase adds little parts to the telomeres of cells when they divide, and those telomeres are necessary to have intact for the next division. So if you keep adding little, they normally get reduced with each division, but if you keep adding segments back on to each end of the chromosome, then you could, in principle, immortalize those cells. So the question is, does Epstein-Barr interfere with telomerase regulation and immortalize the cell? Yes. Does it interfere with apoptosis? Yes. Does it interfere with cell adhesion? Yes. So here's a virus that's associated with cancer that breaks through 4 critical cancer barriers, and it makes sense that viruses would evolve to do this, especially if the virus is selected to be persistent inside of the person it's infecting. Because, just to reiterate what I said before, if it's inside the cell, it causes the cell to divide, it can divide right along with it. If it keeps the cell so that the cell can't have a cap of the maximum number of divisions, the sky's the limit for the virus replication. If it keeps the cell from committing suicide, those viruses aren't going to be lost when the cell kills itself to kill the virus. And if it blocks cell adhesion, then the viruses, instead of growing in just one spot, can spread throughout the body and grow in many different spots and have greater overall replication. Well, I've taken a lot of time talking about Epstein-Barr virus. I've been holding you in suspense about these other viruses here. Just to alleviate the suspense, let me answer the question of whether these viruses break through these barriers all at once. Every one of them does. Okay, this really changes our view of the role of viruses in oncogenesis. And it changes it in quite a fundamental way. What I didn't say when I was talking about mutations is that the old view that you have accumulation of mutations is associated with a severely sort of restrictive paradox. And what I mean by that is mutations would have to interfere with very particular genes in the genome. And in order to get it would be very unlikely in just a few cells to get all of those genes being mutated without getting additional mutations that makes the cell non-functional. Okay, so that's a very difficult feat to accomplish. It's a very big overall barrier to cancer. Okay, getting all the, let's just think about these four barriers, getting mutations in each one of these genes all in a sequence without getting mutations that destroy the lineage. I mean, if you could get cell cycle, once you get all these things broken down, then you can, or at least the first three, cell cycle arrest, immortalization, and apoptosis. Once you get those broken down, then those cells can grow to large numbers, millions, maybe billions of progeny. And then you could get any other mutations you need. If you get a mutation that causes a cell to be non-functional, too bad. You've lost one cell out of millions. So now the numbers are working in your favor if you could actually get all of those barriers blocked at once. And look at what the table tells us. It tells us that the viruses, as a rule, evolve, at least according to these five that have been best studied, as a rule, evolve to block all of those barriers all at once. So that negates that fundamental paradox that's just swept under the rug, or it has been swept under the rug in terms of the feasibility of having mutation only as the model for oncogenesis. Okay, so the question is, that makes sense, but can you really test that idea? Well, there are some very interesting predictions that arise from this interpretation of the data. Remember, we're talking about genetic predispositions plus infection as being sort of initiating steps, and then mutation finishing the job. Okay, and one thing I didn't mention is that though the compromising of those barriers by the viruses is almost never complete, at least so far as we understand it. They're compromised, but they're not completely wiped out. So once you've compromised those barriers and you get cells that are growing to large numbers, large populations of these aberrantly growing cells, mutations that then wipe out that gene altogether, the gene function altogether, not just sort of suppress it, could make those cells grow even more quickly. So that's why, even though the viruses would be selected, if they're selected to be persistent, why they'd be selected to compromise those barriers, that's why mutations in those genes are still going to be likely to occur, and in fact, mutations in a lot of other genes as these cells are dividing. Other genes that might, for example, control cell division. So this argument leads to this very interesting prediction, I think, that's been overlooked, and that is that it's viruses that start the process. If we look at very early stages of oncogenesis, we should see viruses there before we see mutations. It's a very clear prediction. A little hard to test. But you know, it's actually been tested with cervical cancer, the one that's caused by these serotypes of human papillomaviruses, and it's true. Okay, so that gives us some confidence that this actually is the case for these cancers that we know are caused by infections. You get the infection first, and then you get these other mutations that you see abundantly in late-stage cancer. You have them occurring later. But we already accept that cervical cancer is caused by human papillomavirus. I think the importance of this prediction is in its application to those cancers that people are still arguing about in terms of causation. So for cervical cancer, yes. Let's look at another cancer that's a terribly important cancer in humans, breast cancer, and ask the question, do the oncogenic mutations occur later than the infection in breast cancer? Okay, and the answer is, if we knew that for sure, we'd already agree that breast cancer was caused by infections. But let me give a little bit of background on the current state of thinking in terms of breast cancer. First, I want to emphasize that breast cancer is a really important cancer in terms of death, as well as other things that aren't incorporated into a death figure. So here I've got the 5 leading causes of death worldwide from cancers of various sorts. So lung cancer is the leading cause, stomach cancer is the next, liver cancer the next, colon cancer the next, and then breast cancer is next. So 500,000 people a year die, and a lot of other people are severely affected, and families of people are severely affected when an individual has breast cancer. Okay, so let's take a look at what evidence there is relating to the role of viruses in particular in causing breast cancer. Okay, there are 3 viruses that have been associated with breast cancer. One is mouse mammary tumor virus, or a virus very similar to your standard mouse mammary tumor virus. Well, your standard mouse mammary tumor virus, MMTV, causes mammary cancers in the domestic mouse, Mus domesticus. It is found in about 30 to 40% of human breast cancer, but less than 5% of the normal tissue that's adjacent to the cancer tissue in the women who've been tested. Okay, so based on that figure, if MMTV is actually causing the cancer, then we're talking about maybe something around 1/3 of human breast cancer being caused by MMTV. And this now has been supported by several different studies. I've listed some of the references here. Interestingly, when you look at something like breast cancer and you see geographic distributions, the immediate thing that people do, because we're people and our mind is functioning in a certain way, is to sort of gravitate to the things that are being put inside of people. So we sort of think about what kind of dietary constituents could be influencing breast cancer. And that's what happened when people saw that there was a geographic variation in breast cancer. People were looking at the Japanese diet, and they said, well, if breast cancer is less common in Japan, it must be something to do with the diet. Same thing for China. Well, that kind of study led people to conclude there must definitely be some dietary influences. And for a long time, people think high-fat diets, but now the studies that have really looked at fat reduction don't find any compelling evidence for that. Interestingly, if you start thinking about alternative hypotheses like MMTV causing breast cancer, then immediately some alternative tests become apparent. And one of the tests was just published around in 2000 by a group that looked to see whether the distribution of the host of MMTV was correlated with elevated breast cancer rates. And they found that it was. When you're in an area that has this Mus domesticus host of MMTV, your breast cancer rates were about 1/3 higher than neighboring areas in which Mus domesticus wasn't present. So this is another example of how it's very important to keep the hypotheses on the table so that you don't start assuming something's true when you really haven't tested it adequately against alternative hypotheses. MMTV does dysregulate cell cycle arrest like these other viruses, also dysregulates telomerase regulation, so it tends to mortalize the cells. It also dysregulates adhesion molecules. And I've looked, I haven't found any study that looks at the effects of MMTV on apoptosis, so I can't say whether it does or doesn't affect apoptosis. Let's look at the second virus, Epstein-Barr virus, the one we've talked about before. It has been found to occur in about half of the breast cancer specimens in less than 10% of normal adjacent tissue. So again, it's sort of around 30 to 40% of the cancers relative to background. And another study looking at a much broader range outside of the U.S., that first study was from the U.S., another study shown that in several countries, France, Denmark, Netherlands, France, Algiers, Tunisia, find the same tendency for Epstein-Barr to be present in breast cancer at about 1/3 to 40% more than it's present in adjacent tissues. Interestingly, it's more strongly associated with breast cancer when the Epstein-Barr has caused florid infections. And this typically in places like the United States is the case because people are getting infected during their adolescent years, young adult years, and for reasons that aren't quite understood yet, Epstein-Barr causes more florid infections when you're getting infected late. So, by the way, what I'm talking about is also an alternative explanation to what many of you may have heard about that relates breast cancer to the number of cycles. The sort of standard dogma in evolutionary medicine is that people in the United States have elevated risks of breast cancer because they're using birth control, so you have many more cycles, whereas in other hunter-gatherer societies, for example, in other places, women are pregnant more, and so they have fewer cycles. And so that's actually become dogma. But I've looked at the evidence. The data for this are really shaky. In fact, some of the data really don't even exist. It's sort of a speculation somebody made and it got perpetuated. But to make a long story short, when you consider the evidence that people muster for that idea, many of those trends are explained equally well by infection. That is, for example, if women are using birth control, they may be more likely to have sexual partners, they may be getting sexually transmitted pathogens and so on. And some of the trends aren't explained well at all by the idea that you've got more cycling. For example, women have more breast cancer during pregnancy and during the year after pregnancy. And this is just what you expect from infection because your immune system is suppressed during pregnancy, and so viruses that might be causing cancer could get a foothold. Anyhow, that's a whole nother lecture, but since this is a group that's interested in evolutionary medicine, I feel compelled to mention that you can't believe everything you hear in the literature in evolutionary medicine, even though that's the standard, one of the standard arguments people raise. I think it may be partly true, but I think the data favoring infectious causation is far greater. Far more reliable than a lot of the evidence that's been mustered in favor of that hypothesis. Okay, let's look at the third virus, human papillomavirus. This is the same serotypes that are responsible for causing cervical cancer, that also cause head and neck cancer, almost certainly as a result of oral sex. When the person performing the oral sex has a partner who has a urogenital human papillomavirus infection. That's just, I'm mentioning this right halfway through because for college students this is where you start fading and this sort of perks people up. You know, say, yeah, oral sex, watch out. Tonsillar cancer. Okay, good, you're perked up. Good, I was a little worried. Oncogenic HPV are less strongly associated with breast cancers, about 16% of breast cancers in one study, about 25% in another study. Interestingly, if you look at those people who are HPV positive in breast tissue with breast cancer and whether they had cervical cancer, you find that even though the sample size is fairly small here, you find that there's a strong association for people to have HPV positive breast cancer after they have cervical cancer, 46% of 41, whereas none of the 9 cases were positive for HPV when the breast cancer occurred before the cervical cancer. So this strongly suggests that there's contamination of the breast tissue from the urogenital tract, and it's not clear how that contamination occurs. It could be self-contamination by hand, for example, hand to breast contact. Could be contamination during, sex, from genital to oral to breast contact. It could be through the blood or the lymph. So it's not clear exactly what's going on, but the important point is that when you broaden the scope, you start seeing connections that are a little bit worrisome, right? They're a little scary, especially for college students, but not for me, because I'm old. But they're a little scary, but on the other hand, I find them actually very encouraging because if these infections are playing a role, these are the kinds of things we can prevent. And for HPV, we already have a vaccine that'll guard against HPV. So it's just one more, actually I've given you 2 more reasons that probably why you want to have the Gardasil vaccine or any other HPV vaccines that come out. One is cervical cancer, one is head and neck cancer, which has been accepted as caused by these HPV serotypes, and then a third is breast cancer, which hasn't been accepted yet, but it's starting to look as though HPV is playing a role. Okay, so where do we put breast cancer? I'd put it with a question mark. I'd put it based on available evidence, sort of where I put cervical cancer. It looks like infections are playing a role, with these 3 being the big players, and then non-infectious environmental factors playing a role, because the infections alone don't seem enough to do it. You got to have these mutations. Okay, so let's just get back to that question about the prediction of which comes first, the infection or the mutation. Breast cancers have for a long time have been recognized as being associated with genetic predispositions, and one of them is the BRCA1 mutations. This is mutations in the BRCA1 gene, and that gene facilitates the repair of double-strand breaks in DNA, and then it also restricts division, cellular division. So if you have a mutation, you can't repair the double-strand breaks, and you don't have as good control of the cell division. And one of the other big factors, genetic factors in breast cancer was fairly recently discovered to be this other gene called PTEN, which stands for phosphatase intensin homolog gene. And normally PTEN restricts cell division and fosters apoptosis. So if you have a mutation in PTEN, that could lead to greater cell division and less ability for the cells to kill themselves once they recognize that there's something going wrong. So when people saw these two things, they were thinking maybe PTEN is really part of that holy grail for understanding breast cancer, which would be that you've got genetic predisposition, maybe you've got the first mutation that is going to lead again and again to breast cancer, because PTEN is quite common in breast cancer specimens. So it's a reasonable idea, but remember this is looking at the mutation model, and looking at the infection model, we would say, no, we expect PTEN to be late. You can look as much as you want, but you're not going to find it as the initiating event in breast cancer, and that's what people found. And this is a little disturbing and frustrating, but the infectious causation argument that I've outlined would say that if we're going to continue to be looking for that sort of initiating mutation, we're going to continue to be frustrated because it looks like it's the infections that are unleashing those barriers. Now, for some cell types, those barriers aren't there, and those are going to be the cancers in which we might find just mutations and nothing else, or mutations inherited predispositions, like maybe retinoblastoma. Okay, so this raises a great challenge in my mind to scientists who want to study cancer. So this challenge actually has two parts. The part I've been talking about is to find out whether the oncogenic mutations are absent. And oncogenic viruses are present during the earliest phases of oncogenesis. But there's sort of a tricky second part to this. You know, people have made a lot of progress by just looking to see whether cancers are associated with viruses, but there's been some disagreement. Sometimes you do samples, you find, well, these cancer cells don't have a virus. And so you might say, well, maybe the viruses really weren't involved. Maybe it's all confusing. Well, it could be confusing because what we expect to happen as those cancers evolve is that the cancer cells will lose the viruses, because once you've got the viruses setting the process in motion, and once you get mutations in those genes that the viruses have compromised, but now because of those mutations are fully or more fully sabotaged, then the virus is just going to be a monkey on the back of those cancer cells competing with the other cancer cells. So we expect the other cancer cells that don't have the viruses loading down the replication and don't have viruses in cells that can present still some of the remaining viral antigens to the immune system, we expect those viruses that don't have that monkey on the back, that viral monkey on the back, will win out in competition with the cells in the cancer that have the viruses. So we expect, as the cancer progresses, that gradually the viruses will become less functional and maybe lost altogether. And in fact, it looks like we see this in some viral cell lines that are brought into the lab. We see the viruses vanishing from them as they're replicated again and again. Okay, so that's a little tricky. We also have to find out whether oncogenic viruses tend to be lost late in oncogenesis. Well, I think there are some ways around this trickiness. I think there's some new technologies or refinements of technologies that will allow us to use these complicating factors as ways of developing a much more rigorous test. Now, one of the challenges of studying infectious causation of chronic diseases is that you can't use Koch's postulates. There just aren't right models. The infectious processes take too long, and so we have to figure out ways around this. Now, one of the discussants, Dr. Falkow, has been involved in developing molecular techniques to sort of address this problem, really looking at how and when pathogens are associated with diseases. And so that's one way. I think there are going to have to be a lot of different ways that we invoke. The way I want to talk about makes use, again, of molecular techniques, but makes use of what evolutionary biologists called molecular phylogenies. And so we could compare different individuals at different stages of cancer, which is the thing that I talked about with cervical cancer. I said the viruses are there in the beginning, but the mutations are not in the studies that have been done. But I think we could actually generate molecular phylogenies of the cancer in a given individual now, where the people tell me that this is feasible. We can get down to almost single cell level and then amplify the DNA at that single cell level and reconstruct which cells are related to which in the evolutionary process of the cancer within one person, and then reconstruct the evolutionary history just like we would an evolutionary history of mammals or vertebrates, and then get a sense as to what the ancestral type looked like as opposed to the derived types. Remember, the derived types are going to be the ones where we expect all the mutations. The ancestral types are going to be first normal cells and then normal cells that have just viruses. Okay, so this is basically what I'm talking about. This is what an evolutionary tree of a cancer might look like, and the basal lines are the ancestral types that are like your normal cells. We'd expect to find some types that have not evolved very much that would have viruses, intact viruses in them, and then other cells that have evolved a little bit more that have intact viruses. But if we go up in this diagram up to the top, we see the most highly evolved cells. These are the ones that we expect to have high frequency of mutations, both oncogenic mutations and mutations in other genes as well. So that's why I put the yellow arrow there saying increased frequency of oncogenic mutations as we go up to the more highly evolved cells. And these are the ones that are the metastatic cancer cells. And then further right, you see in the turquoise, increasing viral degeneration. So we may find those most highly evolved cells, now that we know which ones are those highly evolved cells based on the molecular phylogeny, we can say these are the cells that we predict will have degenerate viruses or maybe even missing viruses. So now the complication becomes the strength in the testing. So I think this is a way in which we really can go about this. And National Cancer Institute is very interested now, finally after all this time, seeing that some of the greatest advances in controlling cancer have been, really the greatest ones, have been recognizing that viruses are causing cancer, and then you prevent the viruses. You prevent viruses from getting in blood supplies so that people don't get liver cancer. You prevent viruses from infecting people with a vaccine like the Gardasil HPV vaccine. And so what they're thinking now, I think, is that they're likely to go after this idea of infectious causation of cancer much more prospectively. And then they had the last meeting. I was at the first meeting and was giving the main lecture, sort of emphasizing this idea, and I think they're running with it, and they had another meeting 10 days ago, but and they asked me to come back, but I had to give a talk at Cornell that I already committed to, so I don't know what happened there, and I have to check back, but it really looked like they're going to push to try to use this particular kind of technique, as well as other techniques for figuring out whether viruses really are causing which viruses, and then going the next step and trying to prevent those viruses from causing the cancer. Okay, so this takes us back to this quote that I gave you earlier. 1/5 of cancers worldwide are due to chronic infections. The real question is, what causes the other 80%? Well, I talked about the cancers that have been accepted over the last 30 years, and I was saying that I think we're in the middle of this process, of this quiet revolution that I mentioned earlier. And just to sort of give you a sense of my view of this, I now have this table with other cancers that have candidate pathogens, and the evidence is not enough, it's not strong enough to say that these cancers are accepted as being caused by these infectious agents. But the main point for presenting this table is to give you a sense of that we probably are right in the middle of this long process, which for cancer has lasted 30 years already, really more than 30 years. You look at the studies that were occurring before 1980, and I think it's going to last for another 15 or 20 years based on the ideas or based on the progress that had been made in the past and the states of evidence that were available as that progress was being made. So all I've done here is given you some names of these cancers. We've already talked about breast cancer. I've got those 3 viruses. I expect probably between 2010 and 2015, this will now be accepted that some breast cancer, at least, is caused by infections. And I think the HPV data are the shoe-in, because everybody agrees that HPV causes bad cancers. Now that HPV has been found in breast cancers, I think that will sort of break the resistance to accepting this. And then once people accept HPV is associated with breast cancer, then they're, I think, going to be more open to the idea that MMTV and EBV are. I put a question mark there because I don't, and some people will say, well, it's already accepted in the last couple of years that Hodgkin's lymphomas are in part caused by EBV or non-Hodgkin's lymphomas, but I think the majority of people wouldn't say so. So I'm saying, somewhere around 2010, again, this is plus or minus 5 years. Skin cancer is evidence that some are caused by other serotypes of human papillomavirus, maybe 2010 to 2015. Colon cancer, now there have been several studies in the last few years that have again and again supported the presence of this virus, JC virus, which is in a cancer-causing family of viruses, the polyomaviruses. So that now I think is likely to be on the horizon. Merkel cell cancer is a rare cancer that has been associated with a new virus, and I think that's, again, another one that's likely to be accepted fairly soon. Prostate cancer has been associated by actually a Stanford alum with a sort of unusual virus that people had never suspected as being associated with a human cancer, mouse gamma retrovirus, but it's only a small player. The question is are there other viruses? Recently, another polyomavirus, BK virus, has been associated with prostate, and because the polyomaviruses are known as cancer-causing viruses, this is one to look at. Ovarian cancers may also be as well. So I come back to this to mention that a lot is at stake, but if you just look at this table of the cancers that are causing the most mortality, every one of these cancers may be controllable. Lung cancer, preventing smoking; stomach cancer, preventing Helicobacter pylori infection, at least a large portion of stomach cancer; liver cancer by preventing hepatitis viruses; colon cancer, JC virus; breast cancer by these viruses I was talking about. Okay, but that raises another question. If this idea is right, probably a lot of viruses we haven't even identified yet that are maybe contributing to cancers. And so can we make any kind of hypothesis that could be tested that would give us a sense as to what kinds of viruses would be likely to cause cancer? I'm not saying what particular taxonomic group. I'm saying what kind of evolutionary pressures would favor long-term persistence of viruses inside of people, because it's long-term persistence that is associated with breaking down those cancer barriers, which then pushes those cells towards cancer. So the key is to try to understand, why do viruses evolve persistence? And I think there are two general answers. One answer, persistence is probably always pretty good for a pathogen. So if you can evolve persistence, it's, without having some other negative consequence, it's probably in your interest. But there's another answer which I think is very intriguing, another hypothesis, and that is that some modes of transmission of pathogens should have be associated with disproportionately strong selective pressures favoring persistence. And in particular, I think the two, or let's say the one general category of transmission would be transmission by sex, broadly defined to include genital contact as well as kissing. I mean, not just kissing on the lips, but real juicy kind of, sexy kind of kissing. Okay, and here's the logic, is that we don't go around in a given day having those kind of juicy kisses with 20 or 30 people, or having sex, most of us don't, with 20 or 30 people. There are a few professions maybe where people do. But by and large, the opportunities for transmission are very restricted if the pathogen is transmitted by kissing or by sex. So what does that mean? If the next opportunity for the pathogen to be transmitted is 6 months from now or 5 years from now, those pathogens are under incredibly strong selective pressure to be persistent. So even if a pathogen's only been in humans for a fairly short period of time, maybe 10,000 years, that may be a long enough period of time to develop very sophisticated mechanisms for persistence. And when you look at, if you remember that table where I had all the yeses, when you look at the molecular mechanisms, those are very sophisticated mechanisms. There are protein products that are attaching in very specific ways to our cells in order to sabotage those barriers to cancer. Okay, so for a virus especially, but I think also for other pathogens, persistence would be enhanced by having higher proliferation, immortalization of the cells, and inhibition of apoptosis, those three of those barriers I talked about. And so the idea here is not that sexually transmitted pathogens actually benefit from the cancer, but they benefit from the persistence that in effect nudges those cells that are infected towards a more proliferative state, more limitlessly proliferative state, which is pushing them to the brink of cancer. Then if they get the additional mutations, they can go all the way to metastatic cancer. Okay, so let's take a look at some of those cancers that have been associated with viruses and already accepted. And what I've done here is just put in the pathogen, but now I've highlighted the pathogen according to the mode of transmission. So everything under the pathogen column that's in red is a virus that's transmitted by juicy kissing or by genital contact. Okay, and everything that's in turquoise is something that's not transmitted by kissing or by genital contact. Now, for human pathogens, I see some people looking a little worried out there. God, I knew I should not have gone out with that guy. Certainly shouldn't have kissed him on the first date. So you can see there aren't any turquoise there. The average percentage of human pathogens is transmitted by juicy kissing or by genital contacts, about 15 to 20%. Okay, so this is, in this case, it's 100%. Limited, given the sample sizes are small. But we can look at some of the pathogens that are candidate pathogens. We see something, sort of similar. We don't know what the mode of transmission is for a lot of these things. So if the mode of transmission is uncertain, I've just put it in gray. Again, if it's red, it means it's sexually transmitted or kissing transmitted. So, it's at least consistent with the idea that some of these that a disproportionate number of these that we'll find associated with cancers may be pathogens that are transmitted by kissing or by sex. By the way, for prostate cancer, we don't know how they're transmitted, but we do know that men who have had more sexual partners in their past are more likely to have prostate cancer. That creates a strong suggestion that when we find the pathogens if there are pathogens causing prostate cancer, if we find them, some of them at least will be transmitted by sexual contact. Okay, so what's a take-home lesson? Well, I think there are a couple of take-home lessons. One, wait, all right. Okay, no, I didn't mean that. The first take-home lesson, is that we really may be able to do something important as a society about controlling cancer. Because if pathogens are causing cancer, we may be able to eliminate the pathogens and eliminate the cancers. This will be a new golden age of medicine. We've seen nothing like this prior to identifying infectious causes of cancer. So that's the first sort of academic plus, policy, public health type implication. And the second one is the thing you're laughing about, okay? This is something relevant for everybody. If you see somebody, that guy's kind of cute, and you think back about this lecture, okay? Say, not cute enough, okay? But, what we really want is some very simple message. And I thought about simple messages, so I thought about that message. But I don't think that's going to work for this. So I actually been sort of thinking about other messages. So I thought of just say, how much kissing and unprotected sexual contact have you engaged in? Okay, and I think that would be accurate. But again, I maybe not. I don't think it's just not quite short enough. So anyhow, I'm not sure if you have any suggestions for catchy phrases. I'll be happy to hear them. Maybe that's a good place to stop and open the door for the next phase of what we're going to be going into. Okay, thank you.
Thank you for an intriguing and scary summary of the role of microbial replicants on human health. It's a great pleasure and honor to introduce tonight's discussants. For over 2 decades, they've been my mentors, teachers, colleagues, and friends. One of the great joys of academia is the transition from the former to the latter. However, more than ever, I consider them as my teachers and mentors. Dr. Gary Schoolnik is what is referred to in academic medicine as a triple threat. Gifted clinician, outstanding teacher, and insightful researcher. Among other things, I've had the good fortune of having Dr. Schoolnik as one of my attending physicians during my internal medicine rotation. In the field of infectious disease, Dr. Schoolnik's research has focused on a number of areas. However, one of his great loves appears to be diarrhea. I can recall fellow medical students volunteering to be infected with pathogenic strains of E. coli. Now his current work is on cholera, so I thought I'd just throw up John Snow, who figured out the epidemiology of cholera. There's the Broad Street pump. After John Snow figured out the transmission of cholera, he went and had a drink at this pub. Okay, and I don't know what fly atmosphere is, but anyway. Okay, so his current work on cholera has unveiled an epic saga whose scope extends geographically from the highest peaks of the Himalayas to the ocean depths of the Bay of Bengal and across diverse continents. In terms of scale, it extends from the microscopic molecular biology of chitin, the exoskeleton of small crustaceans, to the large-scale behavior of human populations. The work embraces diverse fields, including clinical medicine, global climate change, molecular pathogenesis, population studies, and of course, evolution. Now, Dr. Falkow is a legend in the field of microbiology. He's won numerous awards, and most recently the Lasker Award, often considered the highest accolade in American science. Among other things, Dr. Falkow has pioneered the field of bacterial pathogenesis, how bacteria cause disease in their hosts, including the mechanisms by which bacteria invade cells and the mechanisms used to develop resistance to antibiotics. Some of you might also know him as the person who opens up the first chapter in the book Collapse. He also provided the bacterial strains used in the first recombinant DNA experiments by Stan Cohen and Herb Boyer. More than anything else, those experiments demonstrate the unity of life by showing that even bacteria can correctly express information in human genes. As we heard, he's also developed a modern version of the principles for proving that a pathogen causes disease called molecular Koch's postulates. Now, if Dr. Schoolnik loves diarrhea, then we might say that Dr. Falkow elevates that infatuation to the level of obsession. He may be the only researcher I know who has a collection of coprolites. Those are fossilized animal feces. And he's certainly the only person I know who has one that's been bronzed. As a lecturer, as a lecturer, I have my pin up. As a lecturer, Dr. Falkow was a master at delivering content with humor. As a medical student, I referred to him as the Johnny Carson of diarrhea. You can see why. Subsequently, Dr. Falkow and I shared adjacent office, and I was privy to substantial doses of his wisdom and wit. There are many memes with which he has infected me, including the following phrases. The world is covered with a thin patina of feces. Diarrhea is when stool conforms to the shape of the container, and the goal of a bacterium is to become bacteria. Now, Dr. Falkow is not just a defender of human health, he's also a crusader in the part of bacteria. He's said, I never met a microbe I didn't like, although I'm not sure he said the same thing about people. He also said at dinner that being a pathogen is a noble and honorable profession. Okay. He also helped to teach me how to think like a microbe. Okay, so I just want to point out, he's also an artist. Okay, so he drew that, and then he also, see, we were talking about transmission there. So please join me in welcoming our discussants. Okay. Commentary on the... on the presentation.
I don't know if I should make a rebuttal or a... I won't comment on Paul Ewald's discussion of sex since, like every Stanford professor, those who can't do teach. So,
Thank you. At least I won't get infected.
So, but I did, there's too much really to comment on in Paul's very interesting talk, and so I thought, and since he talked about viruses, I just want to give a small portrait of pylori, but with a little kind of thorn in the end for you. And just to put it in perspective, Helicobacter pylori is a small microorganism. It only has about 1,800 genes in its entire genome that lives in the human stomach. And as near as one can say, they have been with humans as long as there have been humans. Their evolution follows that of language and divisions into human, into different human clades. It is probably at one point in every human being, or very close to being in every human being. Yet it's important to point out that the organism infects, engenders a very good immune response, but still persists for the lifetime of the host in the presence of a very good immune system. But only 15% of people get disease. And of that, probably only 1% get cancer. So it's complex and certainly does depend on the genetics of the host as well as actually the microbe. Now, why is it that Helicobacter pylori is associated with gastric cancer and ulcer disease? And it turns out because it inherited a particular gene, and not all pylori have this gene, some do and some don't. It's called the CAG gene, cancer-associated gene. And the cancer-associated gene is one that is the microorganism has the ability to attach to a gastric cell and then inject into that human cell a protein, the bacterial protein, call it the CAG protein, which is then acted upon by human enzymes, and this sets off a series of events. Now, because I don't want to spend an enormous amount of time on this, I will simply go into two aspects of this. What's in it for the bug? And what's in it for the bug is not to give anyone cancer, even though it fulfills, because of the CAG protein, some of the aspects that you expect of malignancy. It inhibits apoptosis, it affects cell proliferation, and so on. But the immediate effect is to open up the junctions that occur between cells at the epithelial surface and permit the organism to be bathed in nutrients. So the organism injects this protein into a cell for its own good, and that is to be able to get nutrients so it can replicate in this hostile environment of the stomach. Now, actually, people who have malignancy because of H. pylori, one often finds that they no longer have pylori because a cancerous stomach is not a good place for Helicobacter, so it's not what it really wants to do, and it only does so in a small proportion of the time. So having told you very briefly and what it's in for the bug, is there anything in it for the host? That's the point I want to bring up here in the Darwinian sense. And the answer looks like it may be. So before we understood that H. pylori caused cancer, stomach cancer was already decreasing in the Western world. As you saw, worldwide, it's the second cause of cancer death. At one time, it was number 1. But in the West, it's decreased now till it's about number 4 or 5, I think, something of that nature. And that's because it is transmitted probably by either fecal oral route or perhaps even by vomiting. But it is certainly transmitted from person to person with increased hygiene. We're better off. Antibiotics can eradicate the organism. And you would say, good riddance to H. pylori. Now here's the little stinger in the tail. There is very good epidemiologic data now that indicates that as H. pylori has decreased and stomach cancer has gone down accordingly, esophageal cancer has gone up in an enormous way. Moreover, people who have H. pylori are less likely to have certain autoimmune diseases like asthma or and other things. So, does it play a protective role in the human? Is it not so much a pathogen as actually meant to be part of our indigenous flora and play a role in the evolution of humans? And so when you think about, and as Paul ended, I mean, one of his take-home messages is get rid of the pathogens. And that seems a good thing to do, and certainly Gary Schoolnik will agree, and that's the thrust of medicine. But to take a step back, disease sometimes can be a confounding issue when you're talking about biology. And so these things have to be looked at both sides of the coin, and I will just end my remarks with that.
Well, I believe I'm flanked by two masters, and I've been desperately looking for an opening where I could say at least something intelligent, but it's going to be very hard. As Paul went through his talk, I kept saying, well, wait a minute, that's not quite right, and then 5 minutes later, he took away that criticism. So I'm left with relatively little to say, but I will say a few things by way of emphasis. I want to sound a note of caution about the tendency to try to associate cancer causation with infectious agents. And Paul said this very well, but it went by fairly quickly, that the traditional way of proving an infectious agent as the cause of a disease is something that we in the field called Koch's postulates, which basically means that you are able to isolate the responsible organism in a constant manner from individuals suffering the infectious syndrome, grow it in pure culture, and then re-challenge a susceptible host to produce the same syndrome, and then isolate it once again from that secondarily infected person. And for reasons that Paul actually alluded to, that is very difficult kind of requirement when one's talking about infectious agent causation of cancer. And the reasons are in part that it requires the combination of genetic predisposition, certain environmental causations, as well as the infectious agent in most cases to result in a neoplastic outcome from an encounter with an infectious agent. And more than that, it requires a long period of time until secondary mutations accumulate to the extent that the cancer becomes able to replicate in a sustained way and to leave its original site to metastasize elsewhere in the body. Both things make it very difficult to study. The problem is further confounded by the large numbers of infectious agents, including viruses, that we harbor as either remnants of or actual replicating agents in our genomes or elsewhere, which make it very difficult to make a distinction between association and causation. In the proven examples that Paul gave in his short table, there's really no question that infectious agents are significant causes of those cancers. And I think it behooves anyone interested in the study of this area to look at each of those examples, because the pathway from association to causation was not straightforward in any of those cases. And the individual stories or histories of how each one moved from early association to proven causation is a story in its own that's often may have some general things in common, but has so many unique aspects that it bears careful attention. So I think in addition to, since we can't apply Koch's general postulates, we have to have some other criteria in order not to confuse the literature and ourselves. I think, and Paul alluded to them all. The first is finding that the agent is present before the cancer is. The second is that there be a plausible molecular mechanism by which that agent, in fact, could produce the cancer. And the third, which may not be easy at the start, is that an intervention that eradicates the infection or prevents it in some sort of appropriate matched control with susceptible humans, for example, prevents not only infection but cancer in that group. A good example of that kind of study would be, for example, hepatitis B vaccination human papillomavirus vaccination and the prevention, on the one hand, of liver cancer and on the other of cervical cancer. So I just want to end with one comment about this interesting question about whether breast cancer could be due to human papillomavirus. And I'll just remind you that HPV, the oncogenic types of HPV are proven causal agent in cervical cancer, and in head and neck cancer, the evidence is accumulating and is quite strong. What about mammary cancer? And Paul discussed this and wondered just how in the world could HPV whose reservoir is the female genital tract primarily, although men also carry it for sure in a chronic manner. How could that end up in breast cancer? And so, Paul, I'm going to tell you how that happens. It doesn't involve, in this case, sex, which I know will disappoint you in particular, and the audience. But here's how it happens. One of the commonest ways that HPV is transmitted to an innocent susceptible host is during childbirth. A child that's born through a birth canal in which the mother has chronic HPV infection of her cervix frequently acquires the organism through an oral portal of entry. The organism infects the larynx, and many such children will develop HPV laryngeal infection, which can be severe enough in some cases to produce an obstructive lesion of the larynx requiring ENT intervention to widen the airway. That child, having now been infected with HPV, harboring it in the lower respiratory, or the upper respiratory tract, the larynx in particular, when it nurses the mother. And nursing in the third world can go on until the next child is born almost. We're talking about 2 years of daily or multiple daily exposure to HPV through a nursing child. And such children will grow teeth, as many women here can attest, which is often the time when nursing is discontinued. But not in the third world. And so the presence of HPV and a chewing child while still nursing will inoculate that child's mother. And so we have a scenario which starts with sex, an infected male consort with HPV infection of the penis infects the mother, produces cervical infection, innocent child is born through that infected cervix to infect the mother in the breast.
Okay, and then just to follow up, the evidence. I didn't know that. That's great. If is that a hypothesis?
No, it's true.
Okay, and is the evidence that women will only get breast cancer? I mean, the prediction is they'll only get breast cancer if they've had not only a cervical infection but also a baby. In other words, if you have women who do get breast cancer, HPV positive, didn't have a baby, that would reject that hypothesis. But are those data available, do you think?
No.
Oh,
what
okay.
I just told you is putting together information that everything I told you is true.
Except the causation.
Right. I didn't, and I told you how you can prove it.
Okay.
But I didn't provide proof because actually until this very moment, no one has ever thought of this.
No, that means
But now we had the person think of it, and then we got the critical test.
And that women who don't nurse will not get breast cancer, is that right? Is that part of your wrong hypothesis? Good, okay.
But
It's not wrong, Stan.
well, I would just like to say that I really like that, even though you kind of led me along a little bit on that. Because now we can go to the people who gathered those data and ask, did all those women who had HPV also have a baby before? Because there's a very strong possibility of rejecting the hypothesis, and that's what you want to be able to have.
Exactly.
Yeah. Oh, I did. I wanted to. I essentially agreed with everything that you both said, so I don't have anything to argue about. But I did want to just add one more thing. to what Stan had said, and that is related to the association between Helicobacter and reduced allergy and reduced esophageal cancer. This is sort of it's another version of kind of the hygiene hypothesis in a sense. It's sort of saying that if we clean up our microbial fauna, we may end up having adverse results that we wouldn't foresee because we've sort of evolved to, our physiology, biochemistry and all has evolved in the context of having those microbes present. But what I would, and so I think that is definitely an important point to make, but I would want to take it one step further. I would want to say, okay, so let us understand the mechanism of the inhibition of allergy, and also let's make sure that we understand the mechanism of the increased esophageal cancer. Because if we could get rid of, for the esophageal cancer, if the esophageal cancer, let's say, has a mechanism that involves Helicobacter pylori inhibiting another pathogen that then is involved in esophageal cancer, then we may be better off if we were getting rid of Helicobacter and getting rid of the other pathogen, and we get rid of stomach cancer and peptic ulcers and esophageal cancer. So that would be just one scenario by which, if we now push it to another level of investigation, we could come out with the best world of all. And then with regard to allergy, I would say we really need to understand this mechanism, because one group of people might argue that these pathogens, when they're present, are fine-tuning our immune system so we don't have the allergic over-response. An alternative hypothesis is that these pathogens are immunosuppressive, and they're immunosuppressive because they've evolved that for their own benefit. And if they're immunosuppressive, they may have some effects on our immune system that we find beneficial, but simultaneously could have other immunosuppressive effects that would be damaging. And so that could be the case. If H. pylori is immunosuppressive, it could give us a benefit in terms of reduced allergy, but it could expose us to other infections that would be controlled by the immune suppression. This is also important, I think, when people are talking about the sort of worm therapy for allergies, that we know the worms are immunosuppressive. And so we have to be very careful when we encourage immunosuppressive processes, because we've learned from past history with immunosuppressive agents that sometimes something that looks really good, let's say in terms of allergy, could be devastating in terms of having other persistent infections or even acute infections being uncontrolled and then causing damage even worse than an allergic response. So I sort of see these things as continual iterations of logic, and then let's make sure we understand this system fully, and I imagine you agree completely with that.
Actually, I don't.
No, okay.
I don't agree with that. Since this is Darwin's legacy, I think it's important to point out that the colonization of humans, in fact all mammals, by their microbial flora is not a random event, and it's something that's been finely honed by evolution. One enters the world basically sterile, and the first living creature that any baby encounters is in fact some microorganism that begins the process of colonization. There are literally trillions of microbes that make up the human flora, and as you pointed out, or someone pointed out, we are more microbial than we are human in terms of the number of cells that we have. And this, and many of the organisms like Mycobacterium tuberculosis and Helicobacter pylori, 20% of humans have typhoid, carrier typhoid. You can go on and on and on with some, and when you talk about this in the not that distant past, that was the normal flora of humans. And it evolved that way, and it evolved that way for a number of reasons. Now, what has happened is there has been a change in human behavior, and human behavior has been the cause of a number of infectious incidents in recent time. And my favorite is actually the Legionnaire's disease, because if you look at Legionnaire's disease, this is an organism that lives in fresh water in amoeba. And it's done so for, as near as one can tell, forever. On the other hand, if you ask a question, I ask the medical students, how many of you take a bath daily in a bathtub? And nobody really raises their hands anymore. But if you asked that question some years ago, almost everybody would have raised their hands. We now shower. We have indeed aerosolized the entire world, the supermarkets and everything, and we have introduced this organism that lives in fresh water into lungs of many of people who are to some extent compromised in smoke and so on. And Legionnaire's disease, it's a new opportunity for microbes, and humans are constantly doing this, and we did it with toxic shock syndrome, we've done it, and very likely HIV. So I think that the idea that the elimination of microorganisms and so on, just as a preventative measure, may involve some thought about what the consequences may be, and that in the Darwinian sense, if you will.
Could I just add one more thing to that, because it's a very important point, and I agree completely with that generalization, but I would actually have a slightly different sort of guideline or course of action based on the last point that you made, which is that as an evolutionary biologist, or from principles of evolutionary biology, we realize that our common interest with any organism, whether it's one living in us or on us, or other organisms living outside, free living, is our interests are not likely to be 100% in common. There are a few exceptions to that. The best examples are mutualisms, like between an alga and a fungus, which we call a lichen. But I think that when we're looking at these organisms, we can't. I would argue that we really need, and this is really the last point you're making, we really need to understand in as much detail as possible what those interactions are, so we can identify, of the thousands of organisms living in us and on us, which are the ones that on balance are more damaging to us than they are beneficial, and I think then selective. So I'm not advocating selectively wiping out, all our flora or anything close to that, but I do think that we should be, for Helicobacter in particular, we should take a very careful eye to trying to see what are all the negatives and the positives. We've been focusing mostly on the negatives recently, looking at some of the positives, but I think only after we really look at those and see whether or not we can do something else to compensate for any positives. Some other way of controlling allergies, some other way of eliminating esophageal cancer, then we'll be in a position to say, a very informed position to say, well, should we eliminate Helicobacter or not? And I suspect we'll probably find a lot more negatives of Helicobacter than we know about. And one example that seems to be arising right now is associations between Helicobacter and pregnancy sickness. Pregnancy sickness in its normal sort of mild phase seems to be a way in which we protect our developing embryos from toxins in the environment that cause embryonic damage. But some people have pregnancy sickness nausea so severe that they can die of dehydration. And recent work, although it's still controversial, suggests that particularly the virulent variants, the ones that are CAG positive, are associated with pregnancy sickness. And this may be a way in which they enhance their transmission, as you were saying, through vomit, to get spread around in the environment. And in that case, again, we'd have a negative that people didn't realize, 10 years ago, that could be very important. And again, we'd have to say, well, how does this negative stack up against any of the positives? And can we somehow do something to compensate for any beneficial effects? So I think actually, I come back to, I think we're actually agreeing. And if we could just sit and talk a little bit more, we'd say, yeah.
Oh no,
I don't agree with you on this one.
I raise the issue not because I don't think that we should eradicate disease, but I think it's important, particularly in something that's supposed to be about Darwin's legacy, to talk about the consequences of what happens to this very complex biological question.
Okay, well, the fun continues. I'd like to invite the audience up to the microphones on each side to ask some questions of our speakers. I think it's not that frequent that you sit in a lecture and see the progress of science move forward before your eyes. I thought that was exciting. In the past, I would have said it made a quantum leap forward, but somebody pointed out to me recently that that's the smallest possible increase. Okay.
Question primarily for Paul. A lot of work's going on that show a connection between stem cells and cancer. You know all of this, and you didn't mention it. Is there an implication then from your not mentioning it that stem cells are just an intermediary between the infectious cause and the actual cancer itself?
No, I think stem cells, because they have many of these barriers particularly barriers to proliferate, or barriers to total numbers of divisions, compromise or relaxed. They're already reduced. I think they'll be especially vulnerable to cancers. Now, that still doesn't answer whether infections would or would not be involved, but I think if we find some cancers, we do all the exhaustive studies, we find there is no evidence of infectious causation here. I think it'll be the stem cell cancers that already are well on their way or very close to that brink of causing cancer because they actually are in a more proliferative state. So
the question might be, which comes first, the stem cell or the cancer? Is that what you're saying? It might be a chicken.
Well, in this case, I think it's the stem cell that would come first. If the infection were not involved, and it might not be in some of these stem cell cancers, I would say the stem cell cancer comes first, and then the mutations, and then, sorry, the stem cells come first, and then the mutations, and then the cancer. Yeah? Because I would think the normal stem cell is close to the brink of cancer, closer than other cells. That's the way I would interpret it.
Thank you.
Hey, Bob. First of all, I wanted to thank all of you guys for coming and conducting this wonderful panel. I'm a true to form college student. I have all my notes for this question on my hand. So the first part of the question is, I think Nathan Wolf just came to campus to talk, and he was talking about being a drunk looking under the light for the keys. In some sense, it seems like we're looking for maladies and then trying to look for their cause, where, as I see it, there's bacteria, there's archaea, there's viruses, and then there's eukarya. And it seems like all of those could have the host could not take a hit, the host could take a hit, or it could actually benefit the host. So you get 6 possible different combinations. And I was just wondering, I don't know exactly what my question is, but I guess I'm wondering what your take on that is.
Well, that's addressed to me?
I don't, whoever wants to take that.
Okay, who would like to take it?
I would say that in the study, for example, of microbiology, it was very microbe-oriented for a very long period of time. And that's turned out to be not correct. You can't really study any of these things in a vacuum because they interact. And all of the organisms that you described I mean, we have archaea in our intestinal tract, and we have eukaryotes in terms of protozoans and other things. And in most parts of the world, worms and so on. We are a universe of life that interact with one another. And I think what you pointed out at the end is correct. Sometimes it's beneficial, sometimes it's mutual, sometimes it's not so beneficial. But in the main, it's what life is. Yeah.
Dr. Ewald, this is really personal. In 1966, I was working in Ghana, and once a week, I sent Burkitt's tumor tissue back to NIH. And at that time, in 1966, there was a strong sense that it had a viral origin because of the geographic and altitude distributions. And I'm interested in why that particular tumor has that incidence with the young people and why it moved so rapidly. And is it because of the other organism that was associated with it? Was it a plasmodium, I think?
Yeah, actually, that association with plasmodium in sub-Saharan Africa and other places where Plasmodium falciparum is present has been worked out a little bit better in the last couple of years. And what seems to be happening, and this may only be part of the picture, but what seems to be happening is the Plasmodium facilitates the activity of Epstein-Barr. So it actually makes Epstein-Barr replicate more rapidly. So that seems to be at least part of the reason why Plasmodium falciparum is associated with Burkitt's lymphoma, sort of endemic Burkitt's lymphoma. But interestingly, when you look at some of the non-Hodgkin's lymphomas in places like the United States, you see things that look a lot like Burkitt's lymphoma but without Plasmodium falciparum. And more people are beginning to say, well, this is another form of Burkitt's lymphoma that doesn't require Plasmodium falciparum, and it's associated with things that immunocompromise people to a large extent. And that effect is going to be somewhat similar to the effect of Plasmodium falciparum, which is that if you immunocompromise people, Epstein-Barr can actually grow to a greater extent and then be more likely to push those cells towards cancer.
Hi, you talked about the slow pace of acceptance of the viral theory in various cancers. And I wonder if you attribute part or all of that to resistance, undue resistance on the part of the cancer establishment. The reason I ask it that way is a lot of careers and a lot of money has been made both by individuals and by corporations on the backs of traditional cancer treatments. And if true, this would take cancer from a medical problem to a public health problem, where the money-making schemes are very different. So could you address that question, please?
Actually, I did a chapter in a book that came out a couple years ago called Dangerous Ideas, or What's Your Dangerous Idea? And I talked a little bit about the consequences of identifying infectious causation of chronic diseases like cancer, and it's dangerous because it changes our whole economic structure, particularly of companies that are making a lot of money on treatments that don't cure but actually need to be administered on a prolonged basis. But I think that your point is well taken, and I would go one step further. I would say that as a society we have to understand those solutions that are brought about well by sort of free enterprise system, just sort of going ahead as it proceeds, and those solutions that actually aren't going to be developed and administered well by the sort of free enterprise system. And so if we don't, and when we find those kinds of solutions that aren't going to be associated with a big profit, things like vaccines, for example, aren't associated with a big net profit, whereas something like a statin that somebody has to take every day for the rest of their life is associated with tremendous profit. Okay, so the statins, if we're talking about those kinds of drugs, they can be developed maybe most efficiently by free enterprise. But when we're talking about identifying the causes of chronic disease and preventing those causes, especially when those preventions could involve changing our infrastructure, such as screening bloods, getting good markers and then screening blood supplies, or developing vaccines that could be associated with liability, but still what the net benefit could be tremendously positive in terms of eliminating whole groups of cancers. Then I think we have to have our government recognize that it isn't going to be solved by the free market, and so they have to come in and do something for the general good of the population to generate these solutions, and that's going to require not getting their money being diluted by the solutions that aren't particularly effective, the things that are just more window dressing or more palliative kinds of solutions that maybe are slightly helpful, but somebody's going to have a chronic disease for the rest of their life, taking a medication for the rest of their life, generating a lot of profit. Okay, so I think there's some really big issues we have to address that go to the heart of how we administer our health system, how we invest in our health system, and what kinds of things we can leave to companies that are going to make a profit and be efficient at developing solutions, and what kind of things we just can't. And this goes right back to Adam Smith. Adam Smith, over 200 years ago, said there's certain things that the visible hand won't solve very well, and he talked about things like defense of a country. We can't let that be in the hands of companies that are for profit. We have to have some other agency, such as a government, making the calls on that, because it just isn't amenable to the free enterprise system. So these are just some additional examples that I think that are coming up now that our societies and our science has become much more sophisticated, so that we have new things that entities, whether they're market entities or whether they're, let's say, governmental entities, may be able to do to help solve problems.
Yeah, my question's on just get your comments. There's a lot of talk about climate change right now, and occasionally hear that related to microbes and infectious disease. So is that hype, or are you guys concerned, or?
Well, I'll very quickly, since I've done talking, I'll do it very quickly. I would just say that I think sometimes it's going to exacerbate situations. Sometimes it may ameliorate situations. You know, like for example, if you're changing the distribution of mosquitoes that transmit malaria as climate warms, they can move into new areas and move out of areas that they may not be able to live in because it's too arid, okay? And so I think it's probably going to be a mixed bag, and we just have to study these things very carefully to find out what are the exacerbating effects and what are ameliorating effects.
I would only say that for organisms for which use natural reservoirs as their main habitat, and that would be vector-borne diseases like, as Paul said, malaria, dengue, just to name two important examples, and many waterborne infectious diseases, for example, cholera. I would say climate change is very likely to have an exacerbating effect in ways that are hard to predict but may place additional populations at risk. May change the ecology of the organisms as they live in these natural habitats and predispose them from emerging from those habitats to infect human populations. We really have to watch it carefully and have to engage in sort of proactive surveillance, as well as we all have to become ecologists and understand how these organisms not only live with each other in our own bodies, but in natural habitats, how they associate with all the other constituents of those habitats.
Okay. Yeah. Could I say one thing in response there? Okay, I agree with you, but I just wanted to add that sometimes this can be very surprising. So 20 years ago, when people were beginning to be concerned about this with regard to vector-borne diseases, people were saying, well, dengue is poised to spread up through America as the climate is more favorable for mosquitoes, the Aedes mosquitoes that could transmit dengue. And that seemed very reasonable, but there are other variables involved. And in this case, one of the variables is that on this side of the Mexican border, in Texas, really, we're talking about here on the eastern side, there's a fundamental difference between our society and the society on the Mexican side, and that is that our living is pretty much in mosquito-proof habitats. So we live in air-conditioned cars, we drive a lot in cars, we're living in air-conditioned houses, we spend a lot of time inside in workplace. And although there were terrible outbreaks south of the border where thousands of people were infected, the dengue was sort of out of control, CDCs have done studies in which they've looked at how many new cases are generated from a given case of dengue north of the border, and they found that the number was so small the dengue couldn't maintain itself. So I only mention that because it's a complicated issue, and climate change could have caused that to spread had we not been in more of a mosquito-proof environment. But given that we're in a mosquito-proof environment, that seems to have protected us against the effects that the warming could have had.
Two last questions.
I might have some misinformation, but it's my understanding that women who bear children later in life or don't bear children are at greater risk of breast cancer than those who bear more children younger. I'm not sure if that's right. Then how does that go with the theory that was proposed about how HPV could be transmitted, as well as do you have any theory about the connection with hormone replacement therapy in this whole mix?
Okay, so right, that's one of these associations, and basically the data indicate that if you have a child early in life, that is protective against breast cancer after menopause, but not premenopausal breast cancer, which is sort of weird, right? Why would it be protective against one or not the other? So that's sort of a paradox. What I would argue is those associations are important associations to explain, but we have to be sure to think about all of the hypotheses and ask, are there any hypotheses that are inconsistent with, or is that association inconsistent with any of these hypotheses? And so one of the things that I would certainly want to look at there is how having a baby early on is associated with how many sexual partners a person has later on. Women who have babies, let's say in their 20s, around 20, are generally, historically, they've generally been in relationships with people that in many cases at least, are fairly stable. Women who are in their 20s and are not having kids are maybe not having the motivation to stay in that relationship that you would have if you had a kid with somebody, okay? And so I would want to look to see whether women who had children early had fewer sexual partners during their ensuing 20 years than women who were single, swinging single, whatever you want to call them. I know I'm sort of shaking around here. But I would certainly want to look at those data to find out whether or not there was a difference before I would assume that it was actually the pregnancy that was protective. Because I would want to know, why wouldn't pregnancy protect against premenopausal breast cancer? Why is it only postmenopausal? Whereas if it were that women who were not having kids also were changing partners more frequently over those 20 years, then you might, that could be an explanation for why you would tend to have the accumulation of, let's say, exposure to pathogens causing breast cancer postmenopausally. Now, with hormone replacement therapy, that's an interesting one. For a while, people were thinking hormone replacement therapy actually lowered breast cancer, but it looks now as though, if anything, it's probably slightly elevating breast cancer. Now, the interesting point with the hormonal aspects of breast cancer is that the associations with women, for example, in breast cancer as opposed to men, as well as associations with estrogen and progesterone intake, either whether it's through birth control or hormone replacement therapy, have led people to say that maybe those hormones are directly causing the breast cancer, directly facilitating proliferation. But it's more complicated because the hormones are also immune suppressive. And the reason for that, the evolutionary reason for that, is apparently that if you don't suppress the cell-mediated immunity during those last 2 weeks of every monthly period and during pregnancy, there's a chance that you're going to attack the sperm cells and the zygote and the fetus. And so immune suppression of some parts of the immune system is important. And estrogen and progesterone cause that immune suppression. Okay, so if you're then saying, well, how do we explain the association between estrogen, progesterone, and breast cancer? Then we've got one hypothesis, which is that it's directly proliferative. And then we've got another hypothesis, which is that it's immunosuppressive, and then the pathogens that normally would be kept under control are replicating. So in that case, we don't really have a way of distinguishing between direct effects of estrogen, progesterone, and indirect effects on pathogens through immune suppression. So that's an example where, you could make an argument both ways. But it is a bit surprising. Well, the other issue, when you actually look at the data, the people have been arguing that estrogen progesterone is important for the higher rates of cancer in places like the United States where you have a lot of birth control. The only way they can really make that argument work is that they're saying that it's the proliferative effects when you are taking these compounds early it's the proliferative effects associated with cycling that then increases mutation rates early on, and then that causes cancer postmenopausally, which to me sounds like a big stretch. I mean, I had a hard time presenting it because it seems like it's sort of boxed into a corner. So anyhow, my general answer is let's make sure that we look at all the effects, direct and indirect effects of these hormones before we make the conclusion that it's a direct effect of estrogen on cancer incidence. Oh, the other thing I would add is that, as long as I love to talk, the other thing I'd add is that a lot of these cancers that are associated with elevated estrogen or progesterone are estrogen or progesterone negative, receptor negative. So that's a paradox. Now, there are people who tried to sort of argue around that, but that, to me, argues a bit more in favor of the immune suppressive effects of these hormones rather than the direct effects, because particularly aggressive breast cancers don't have the receptors expressed for these hormones. So how can it be a direct effect?
Okay, well, it's been a fascinating discussion. We've run out of our allocated time. I invite any of you who have additional questions to come down here and talk with our speaker, our panelist. Thank you very much for coming. I'll just close with the words. I believe it was Joshua Lederberg. He says, in the contest between microbes and humans, I'm betting on the microbes. Thank you very much.
Good job.
Thank you.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.